Hello students. So we are going to learn about different gene transfer methods today. As such, there are three different methods that are used for gene transfer from one bacterial cell to another bacterial cell, that is transformation, transduction, and conjugation. In today's lecture, we'll learn about transformation as a gene transfer method. So transformation is a genetic transfer process by which free DNA is incorporated into the recipient cell and it brings about genetic change in the recipient cell. Even within the transformable genera, only few or certain strains or species are transformable, and the ability of these strains to pick up foreign DNA molecule is because of a genetically determined property which is known as competency of the bacteria. Competency in bacteria is a regulated process and it is important for uptake of foreign DNA molecule. Several competent specific proteins include membrane-associated DNA binding protein, a cell wall autolysine, and various nucleases. Quorum sensing is an important pathway that regulates natural competence process in which a regulatory system responds to cell density. So here cells produce and excrete small amount of peptides during the growth. And when these peptides accumulate to high enough concentration, all the cells in the population are induced and they all become competent. This slide talks about the mechanism of DNA uptake during transformation. So during natural transformation, competing bacteria first bind to the DNA reversibly, and after a while, the interaction or the binding becomes completely irreversible. During entry of the DNA into the cell membrane, the membrane-associated nucleases will act on the double-stranded DNA and will start degrading one of the strands, such that only single-stranded DNA is able to enter inside the bacterial cell. As soon as it enters inside the bacterial cell, it is coated by single-stranded DNA binding proteins, which prevent its degradation and stabilizes it. During the last step, recombination reaction is mediated by a Rec-A protein that will lead to generation of transformed bacterial cell. Artificial competence is basically not natural competence, but it is induced in bacteria by treatment with high concentration of calcium and chilling the cells for several minutes. Cells of E. coli that are treated in this manner will take up the double-stranded DNA and therefore they will be transformed at a very high efficiency with the foreign DNA molecule. In general, two methods of artificial competence, that is chemical transformation method and electroporation, are commonly used in laboratories. In chemical transformation method, the chemically competent cells are first mixed and incubated with the foreign DNA molecule, and then they are given a heat shock at about 42 degrees Celsius for 30 seconds. The cells are then transformed back into ice so that they can recover. A recovery medium is also added at this stage, and then the cells are incubated at 37 degrees Celsius for about an hour, allowing the cells to recover completely. After plating, the recovered and the transformed cells can be observed on the Petri plate in the form of colonies. In electroporation method, we make use of an equipment or an instrument which is known as an electroporator. Electroporator has a chamber in which you place a cell which is known as a cuvette. So this is a electroporation cuvette which you can observe. So it basically has the positive and the negative electrodes, and in the space between the two, you add your sample. So your sample in electroporation is basically again a mixture of your electrocompetent cells that are mixed with the DNA, foreign DNA, that is, for example, plasmid DNA. And then the cells are kept on ice for a couple of minutes. First, the cells are mixed and they are placed in the cuvette, and then the cuvette is placed in the ice for a couple of minutes. Followed by this, they are transferred into the this slot. After opening the lid, there is a slot present where we need to keep the cuvette, and we need to give a pulse to it by just pressing the tab in the program, and it will give it a short pulse of about 5 microseconds of about 15 kilovolts per centimeter. This leads to disruption of the cell membrane and entry of the foreign DNA molecule inside the cell. Again, there is a recovery stage where we expose we add media like LB to the cells, and we incubate it at 37 for about an hour. After that, we do the plating, and the recovered cells grow as transformed cells on the medium. This is about transformation. Thank you very much.